Hello, everybody. Today we are going to talk about plant physiology. You may not be super interested in plants. I wasn't when I was an undergrad myself, but the more I have learned about them, the more interesting I have found them. Their structures are simple but elegant. They do amazing things without having a nervous system or a muscle system like animals do. And so I hope by the end of this lecture, you'll be intrigued by plant physiology as I have become and will want to learn more. This is a quick outline. We're going to talk about how plants grow. They go through the growth process and development process very, very different from how animals do it. In animals, it's stereotyped, it's set. They're going to do it exactly the same way from zygote to individual. It's built. And then if anything goes wrong, there isn't a chance most of the time for a do-over. Plants have incredibly flexible and adaptable ability to change how their body is growing throughout their lives and can live indefinitely, some kinds of plants. We're going to look at how they obtain their needed nutrients. It's a little more complicated than it looks like. You don't have a plant who's just sitting there in the soil doing nothing. They're actively managing and monitoring their own body to accomplish homeostasis, like all living things do. We're going to look at how they move substances around in their bodies. They are masters of physics and chemistry. They don't just have a big pump like animals do, squeezing some muscles. They have to use a very sophisticated set of structures, and the way that the water moves through their bodies is absolutely fascinating. We're going to look at how do plants sense and respond to the environment. Now, we've talked about having hormones and a nervous system in animals. Plants do not have a nervous system. They do not have nerve cells, a special tissue type like animals do. They do have hormones. We'll give some examples of the kinds of plant hormone molecules and what they do in plant bodies. We'll also look at some of the responses that plants are able to make. So again, we think of plants as just sitting there doing nothing. It turns out they're doing a lot and that they're able to detect changes or conditions in the environment without having the kinds of sense organs that we think of as doing that job. And that although they can't get up and go somewhere else, they can't do locomotion, they can move and they can change what they do. It may be slow, but the phrase I think of overall is that plants can't go, so they grow. That they will grow towards the resources that they need. So we'll look at some examples of hormones in plants and how they even communicate with one another and with other species. These are active areas of research. We have been just scratching the surface on how plants use hormones for communication and how they're, excuse me, how they interact with other plants through the soil. It's a fascinating area of active current research. All right, we're going to start with the birth of a new plant. We talked about the idea of what a seed is in lab already this semester. So some of what we'll be talking about in this lecture, we've already talked about in lab. So this will be review, we'll be reinforcing and expanding upon some of those concepts. So we have a seed. This is an example of a bean seed. So it's a dicot. And the seed will be under a little bit of soil. Often it needs still to be able to detect sunlight, and it will need water. And once it has detected those things and also a temperature that is appropriate for it to survive in, those will trigger a process called germination. Germination starts with a set of chemical reactions that mobilize the resources that are stored in the seed and trigger the beginning of growth of the embryo. It will start growing roots. It will start growing a shoot, and the roots will go down. We'll talk about how they know what direction to grow in a little while. We talk about hormones. The roots will grow down. The stem will start to grow up, and then the very first structures that we see above ground, besides the stem, are the cotyledons. The cotyledons are the embryonic leaves. They existed in the embryo that was inside the seed, but once the seed germinates, it will very rapidly, using stored resources that are often in the actual cotyledons, they will sort of shrink up the cotyledons, the embryonic leaves, and grow the first true leaves or foliage leaves. All right, so remember that those descriptions, monocot and dicot, of whether our plants belong into one taxonomic group versus another. The first reason we can tell whether plants belong in that one of those groups or the other is when they sprout up, when they germinate from the ground. Do we get one little leaf sticking up like a corn plant or a palm tree, or do we get two little leaves sticking up like a bean plant or a basil plant or hundreds of thousands of other kinds of plants? So another important process that's part of germination, remember the structure of the seed includes the embryo, those stored resources, and that hard protective coating around the outside. So part of germination is also absorbing enough water that it literally allows them to sort of crack out of the seed coat to be able to start to grow. It's kind of like hatching out of the egg for a bird or a reptile. All right, we also talked about the idea of meristems in lab earlier in the semester. These are regions of the plant where throughout its life, it will have stem cells. Stem cells are cells that are able to divide and then differentiate, that is, turn into any kind of cell that the body needs. So in animals, most animals, we only have stem cells for some tissue types. For example, we have blood stem cells in our bone marrow that function throughout our lives, but most types of cells, we don't have a chance to have new ones formed and differentiated and grow into a particular tissue after a certain set point in development has passed. So research into stem cells for human biology is because we want to be able to have medical technology that allows us to repair damage to body organs, especially in the nervous system, later in life when we have had injuries or damage to them. Plants have these meristematic regions where throughout their entire life, the cells are going to be able to divide and differentiate into any kind of cell that is needed. Again, we'll talk later about hormonal control over the process, which determines what the cells, how the cells know what kind of cell to turn into. So when the plant, the baby plant, is in its first year, some plants don't live more than a year. Often these are small, predominantly leafy, flowering plants. And back at the beginning of the semester, we talked about different body forms of plants. So these would be the kind we call forbs, F-O-R-B-S. And also grasses fall into this category. That's different from shrubs and trees that are going to grow more than 1 year. After the first year growth, they're going to start producing that brownish colored, harder structure we call wood. But when we have those plants still growing in their first year, they're only going to have green growth. So this is called primary growth. And that means first, we'll have secondary growth later. Primary growth is going to be on the north-south axis of the body only. So we have a root, tip, meristem. We have a shoot, tip, meristem. And those are going to allow growth to occur up and down. Right. Because those occur at the tips, those are going to be referred to as our apical. Apex is the tip of the mountain. Apical meristems. We'll have another kind of meristem in the next slide. So we want to distinguish that. Now, growth at the apical meristems is also going to continue throughout life. It's the only kind of growth that's happening in the first year. There's 3 steps to how plants grow in their first year. And the first step is cell division. Right, so they're going to be doing mitosis, and the mitosis is going to be happening right there in the meristem. So the meristem is going to be down near the tip. It's protected by this cap of sturdier, more reinforced with additional cellulose. As the root is trying to grow down into the soil, soil is abrasive, right? So in order to protect the delicate cells of the meristem, as the root is pushing down into the soil, it will have some abrasion, and this root cap protects it. It actually also secretes mucus to sort of ease its way into the soil. So cells are dividing here. So what we're going to see is that we have a plant cell with its nucleus, and what's going to happen next is that single plant cell going to build a new cell wall, and so that it has 2 nuclei. It's now just thrown up a partition of cell wall in between them, and then it's going to go into this zone of elongation. So the root is going to be growing behind the tip. And what happens in this elongation is cell growth. And so what actually happens is the cells themselves are able to expand in size. So we start with these smaller cells that are produced by mitosis. And then what happens is each of the new cells then gets longer and skinnier. So the cells elongate. And the reason they can do this is that the structure of the cell wall is wrapped in a spiral that is perpendicular to the axis of cell division. And as they are filling up with water, the water actually stretches the cell wall surrounding the nucleus. So we see very rapid growth. So an increase in size of those cells after they have been generated. Also not something in animal cells. All right, so we have the zone of cell division where mitosis happens, we get more cells. The zone of elongation where cell stretching happens, and we don't get more cells, but the cells themselves get bigger. And then once a little more time has passed, then the cells are in this area where once they've sort of grown in size, then they start to turn into the correct kind of cells for that point in the plant body. So differentiation is a word we've already used in animal development and also in the concept of a multicellular organism. Our definition of a multicellular organism has 2 parts. One is that it has multiple cells, more than one, many, and that those cells have to work together to be integrated. That's one part. The other part of our definition is that there have to be different kinds of cells that do different jobs. So the zone of differentiation is where the cells start to grow the structures that are going to be important for that body part. So for example, this is a root meristem. So what will start happening in the zone of differentiation is that the vascular cylinder will be established. Notice that the vascular cylinder cells are not very different from the other cells when we get closer to the meristem. And also that it will start growing root hairs out to the side. So those root hairs are going to be some of those little wiggly extensions like we talked about that increase the surface area of a structure. The main jobs of the root are to structurally support the plant and hold it up, make sure it's well physically established in its location, it doesn't tip over, and also absorption from the soil of both water and nutrients. Just like a lung has an extremely convoluted internal structure to increase the surface area, these little root hairs extending out away from the root itself increase the total surface area. That not only increases friction to help provide structural stability, but it also increases the surface area for absorption, right? Because absorption is physiologically the primary job of the root. All right, so if we have a kind of plant that is going to live more than 1 year, it is only growing up and down in the first year, but when it gets into its second year, it is going to start to be able to grow. We have two directions of growth in plants. We have up and down and we have out, right? So it also has two kinds of meristems that are responsible for secondary growth, right? Primary growth is going to happen in the first year. Our basic plant stem, plant root that we've looked at in lab already are going to be what those plant cross sections of plant stem and root look like in its first year. In dicots, there's going to be this ring of the vascular bundles. But if it's going to become a much larger plant, it actually needs to have a bigger volume or proportion of its internal cross section eventually filling up with more vascular tissue. So it needs to be able to grow the vascular tissue, and it also needs to grow that protective woody outer structure. So usually we say the tree has bark, B-A-R-K, and that's that brownish, sort of almost scaly looking sometimes, really sturdy, hard structure that's wrapped around the outside. It's like our skin. It's going to protect the plant from physical harm, from pathogens, from parasites, and also be very, very waterproof. It also provides structural support because it's made of these very hard cell wall material that only grows in secondary growth. All right, so we have a type of meristem. Now these lateral meristems have a different name. They're called cambium. All right, so we have vascular cambium, right? And it's actually a tube, right? So the actual, instead of being a ball like the apical meristems are, the cambia are actually a ring of cells, and they go down through the whole stem. So it's a cylinder of cells. The vascular cambium is going to grow new vascular tissue. The cork cambium is close to the outside of the diameter. It's also a ring or a cylinder, and it's going to grow this outer structure, this sort of skin-like structure that's called the periderm. Right? We have an epidermis, right, outside structure of our bodies. So plants are going to have this periderm. And it's produced, right? It's outside of this cork cambium. And so the cork cambium is going to only do cell division on one side, and it's going to produce the periderm or the bark on the outside of that. The vascular cambium actually forms a ring that is in between the xylem and the phloem. And so it's going to be growing secondary xylem towards the center of the ring of the plant stem, and it's going to be growing phloem towards the outside of that ring, right? So instead of having these little separate vascular bundles in our plant stem after the first year, we start to see that the vascular tissue forms a continuous ring. It's more like a layer rather than these separate bundles, right? So we continue to grow both additional xylem and additional phloem as the plant becomes larger and larger in diameter. So the cell walls of the plant cells that are grown in secondary growth not only contain the cellulose molecule that we already talked about making up cell walls, but they have a different molecule that's even harder and sturdier, and it's called lignin. All right, so lignin is a molecule that can be used in secondary cell walls, and that it produces the material that we refer to as wood. So as the plant is growing, slight differences in how much it grows during winter versus summer are recorded in the history of the growth of its secondary xylem and phloem. And so when we slice through a tree, a larger plant that has grown for many years, we actually can see the tree rings, we usually refer to them as. And these are shown as secondary xylem cells. As the plant continues growing in diameter, it sort of abandons these older cells, it leaves them behind, it keeps growing more cells, they're more dense during the rapid growth periods of time, and they're less dense during less rapid periods of time. So sort of the thickness of those growth rings and the thickness of the spaces in between them can tell us about the history of the plant. Now, we don't anymore like to go around cutting trees down generally in order just to look at the ecological histories of that forest. So if a tree falls over, especially a very large old one, tree scientists are running out there, slicing it up, looking at it. We have sliced tree rings from very old trees historically, and we can look at their growth rings and we can correlate them to periods of time in the environment, which were rainy and there were thicker growth bands in the tree because it had plenty of water and it was growing faster, and periods of time when growth was slower. So individuals also may have personal things that are happening in their history that we can also see in the tree ring. So people who do this work are called dendrochronologists, and they study the timeline of the lives of trees using tree rings. We can see in this tree, for example, that the middle of it is kind of offset from center. So it actually had something pushing on it at some point during its life. If you look at picture C, another tree fell and made it lean over a little bit. So it was growing faster on one side than the other and sort of to straighten itself out. We can see a history of fire here on part F, and that band is thicker as the tree was trying to regrow its bark in order to protect itself. We can see damage possibly from bark beetles that the tree is vulnerable to when it has undergone a stressful period of time, usually for trees meaning drought. A modern technology to help us study the history of trees is, instead of having to cut the tree down, we can take a core. So just like when we go into ice or when we go into sediment, and we sort of force into that structure, that material, we sort of force in a tube, a small tube. We can take sort of a drill bit that's a long tube that has a sharp end and it's rotating, and as it cuts into the tree, it makes a little tube, it makes a little column of the material. We can drill that into a large tree, take that core out and we get just a spoke from its history. So we don't have to kill the tree. It'll be able to, we put a Band-Aid on the outside of that hole more or less, and it doesn't harm the tree. It can recover from that, just like when you get a biopsy or a blood sample taken. Right, as we start looking at how plants grow, a really important thing to consider, if you thought plants were boring, soil. Often we don't think too much about soil. We might think, oh, it's just under our feet. Not much is going on down there. You are wrong, my friends. Okay, so a whole lot is going on with soil. And the plants that are growing in it depend on it for the water and nutrients that they need. Soil is actually a complex system. It isn't a static system. Soil is continuously developing, and we're getting more of it. And also we're losing some from the surface of our terrestrial ecosystems as it gets worn away by wind, by water. So topsoil, or this soil that's on the very top of the layer, if it doesn't have quite a lot of plants growing on it, maybe some kind of mulching material on top, dead leaves or piles of rocks or cobbles, that it's susceptible to getting blown or washed or worn away. The very deepest layer underneath the soil is going to be solid rock, and solid rock is going to eventually erode and weather, like we talked about as a biogeochemical process. It's going to get disintegrated basically, and then it turns into coarse grained rock. And then eventually it gets mixed in with more biological components and water and gets broken down even more into smaller particles. But soil itself is made up of tiny fragments of rock mixed in with a bunch of other things. So what are some of the things? Right, this top layer that looks dark gray or brown, it's moist. The deeper we go down the soil, the more rock material it is, the less it's going to be able to retain water. So the plant roots are only growing in this top part, the horizon A. Soil levels are called horizons, and they're defined by how much organic matter and the structure of the rock material that's in them. Fascinating stuff. Take a geology class if you're interested more in the components and makeup of soil. So depending on the mineral composition of the rock that is in this geographic location, the mineral composition of the soil can be chemically different. So if we have a kind of rock that has particular structure made because of the way that it was formed underlying the soil in a geographic location, the soil itself is going to have different mineral nutrients that are present in it. It may have a different pH. It may have certain materials that are part of the soil that are different in different geographic locations because of the mineral composition of the rock. So we actually see superimposed across some different ecosystems, little pockets or little areas that, because of the geologic history, have a different soil type. They may have dramatically different plants growing on them. There's a kind of soil in some areas of California called serpentine, and it grows on outcroppings of this serpentine rock. And there are plant specialists that grow on those locations. So we could be walking around a landscape and we could see sort of an abrupt transition from one kind of plant being there. And that tells us that there's a special habitat, a patch of special habitat there. The other things that are mixed into the soil to make it soil rather than rock include water. They include dead and decaying organic matter, such as leaf particles, dead animal particles. And then there's actually quite a tremendous amount of bacteria and fungi that are living in the soil. Remember that fungi can have this extensive network of cells Underground, we don't see them, we don't think about them. But if we actually sort of slice in and look at a three-dimensional image of what exists underground, we have this network of fungal hyphae. It is going to be intertwined with the roots of the living plants that are there. There may be decaying plant roots of plants that were at the surface that died. And then there's a whole community of not just bacteria and fungi, but soil organisms ranging from single-celled eukaryotic organisms to microscopic and macroscopic organisms. So we'll have a whole array of nematode worms, of annelid worms like earthworms. We'll have soil-dwelling arthropods, a variety of different insects. The larger animals are not living in between those soil particles, but they may be burrowing, right, as they move through the soil and dig those burrows or make those tunnels. As an earthworm feeds, it is eating soil and making an earthworm-sized tunnel behind it as it goes along. That mixing of the soil that animal activity does also provides a way for oxygen to get down into the soil as well. So the animal community is incredibly important in forming the structure of the soil, and that the plant roots depend on all of those things happening. So the amount of organic matter that is in the soil acts like a sponge. And so where we have sort of fluffier, like darker colored soil, that's showing there's a lot of organic material, those little fragments and crumbs of decayed leaves and other plant matter and of dead organisms, provides a way for water to stay suspended and around the soil particles. And that's critically important. If we don't have this sort of spongy quality of the organic matter, the plant roots can't grow there. So when we want to improve the quality or the production of the vegetables in our vegetable garden, what do? We put a lot of compost and we put mulch onto our soil. That helps build the organic structure of the soil, helps it retain water, and adds nutrients to the soil. Where we have areas that have previously been used for agriculture for a long period of time, modern agricultural practices where we have one kind of plant growing, say corn, it grows through a part of the year, and then we chop down all of that plant matter that's left. If we were to clear that plant matter away, we just have bare soil sitting there. During the time the soil is bare, it is subject to erosion and removal, right? So farming practices that don't let soil lay fallow, which is not being used, or that leave the dead plant matter, or even better, till it, cut it or shred it up and mix it down into the soil to help replenish those nutrients. Those are practices that can help preserve the topsoil, right? Once that topsoil's gone, agriculture may be impossible in that area. And topsoil grows or accumulates very, very slowly, less than a centimeter per year. So in areas where that have had farming practices that tend to deplete the soil or that dry it out, we can actually lose the ability of that soil to produce crops over long periods of time. So different kinds of structures of soils exist in different biomes, partly depending on the kinds of plants that live there and the turnover in their, how much stuff falls on the ground and how many nutrients and what kinds of nutrients. The plants that grow in each of those biomes are adapted to those conditions. So here in Southern California, we have these chaparral plants. Well, the native soil that is in our area is a sedimentary rock that was formed. We had the flat areas around San Diego were ancient floodplains and shallow coastal regions. So a lot of sediment was washed down from the mountains in rivers to our east. It flowed into these flat coastal plains, piled up, has a lot of rocks in it, and they're round and smooth because they tumbled down from those mountains and got sort of polished and smoothed off. And so they're mixed in with our soil, and our soil is not, it doesn't hold very much water. It's kind of chalky and drier, and it doesn't have that many nutrients because it wasn't formed by a whole bunch of old forests piling and piling and piling up on top of one another. The native plants in our area are adapted to that. They grow best, and they're happy in dry-ish, nutrient-poor soil. If you take a white sage plant or a chemise or other chaparral plants, and you put them in a vegetable garden with really moist soil and with lots of nutrients, they're probably just going to die because their roots are going to rot. They won't be happy in that environment. Right. So even though what we would consider the quality of the soil, the characteristics of it are going to be different in different environments, the plants that are native to that environment, that have adapted with it, have co-evolved in that ecosystem for thousands or tens of thousands of years, that's normal for them. That's what they're going to have, structures that are adapted, functions that are adapted to survive in those environments. All right, so let's think about soil on a little more zoomed in scale. Soil isn't going to exist in a completely dry environment. So actually, if you imagine you put a bunch of marbles into a big beaker and then you pour water, right? The water is going to be filling up all the spaces in between the soil particles. The soil particles are the marbles. So we're going to have this water that's existing in between the soil particles. This also happens in sediment as well. So the water that kind of sinks down in between the rock particles in sediment is called pore water. And we have to think about how the roots of the plant are going to interact with the soil particles, that they're both surrounded by this watery environment. Turns out, because of the chemistry of rocks, that they are actually going to have a slight negative charge at their surface. So any of the dissolved ions that are positive, that are in that soil pore water, are actually going to tend to stick to those rock particles like magnets. A plant wants to be able to absorb those mineral nutrients, and so it has to have some way of getting those positive ions unstuck from the soil particles. It so happens that the plant root is going to be doing not photosynthesis, because it's underground and it doesn't have any sunlight. And if you pull plant roots up from the ground, you see that they aren't green, they're whitish or brown or sort of translucent. So they're not doing photosynthesis. So the predominant cellular function is going to be cellular respiration. So the plants are going to actually need to absorb oxygen from the soil water around them, which is why we talked about having animals moving around in the soil and mixing it up and making little air spaces in it, is going to make it possible for the oxygen to get down to the roots. But they're also going to be secreting carbon dioxide as a waste product, right? So they're going to be exhaling carbon dioxide through their roots. What happens to carbon dioxide when it mixes into a watery solution is that it goes through a reversible chemical reaction and is going to convert into bicarbonate ion. Bicarbonate ion is weakly acidic, and so we're going to get hydrogen ions that are freed from the molecule and are going to actually stick to the soil particles. So there's also additional hydrogen ions that plants may generate from other biological functions, right? So they're going to secrete some of these hydrogen ions. So we have both of these examples on this slide. We have release of carbon dioxide and water that turns into bicarbonate ion plus hydrogen ion. We also have secretion of hydrogen ion, any kinds of molecular processes where the plant may be breaking down one kind of molecule, turning it into another. It may release hydrogen ions. It doesn't want its body fluids to be too acidic, so it will excrete excess hydrogen ions. This happens to help it be able to absorb positive ions from the soil solution. And that happens because those hydrogen ions get stuck to the negative soil particles and it bumps off the positive ions that the plant wants to absorb. So there's kind of this competition between the hydrogen ions and any other kinds of positive ions by the plant roots actively making the soil solution a little acidic. It actually releases those other cations, the positive ions that are mineral nutrients that the plant needs. When they're not stuck onto the soil particles, they're actually going to be able to be absorbed by the plant, right? So it frees them up into the solution of the water that's in the soil, and then we can see the plant is able to absorb them. Right, the plant may be doing active transport to pump those other ions in as long as they are there and they're available. All right, so when we're thinking about plant nutrients and plant nutrition, we want to touch on what a plants need, right? They need to have carbon dioxide and sunlight coming into the leaves for them to do photosynthesis. They need to have water coming in from the soil. But when we say plants need nutrients, what do we mean? There's two categories of plant nutrients. They're categorized as macronutrients, ones that plant needs a lot of, and micronutrients, ones that they don't need that much of. All right, so this slide is showing what happens when we have a healthy plant compared to when we have a plant that's deficient or hasn't been getting enough nutrients of its three main macronutrients, right? The macronutrients for plants, and if you go to a garden center and you get some fertilizer for your plants, it's going to say how much of these different three nutrients it has, right? So we can think about this as NPK. We have nitrogen, we have phosphate, the P, and potassium, the K. They'll show these symptoms if they have a deficiency in any of these nutrients. And if we see this sort of, there's other ways the plant leaves will look if they have other things like an iron deficiency. So these are the macronutrients, ones they need lots of, nitrogen, potassium, and phosphate. The micronutrients, so we need micronutrients too, and a lot of the ones that we need participate as vitamins in, or they're needed to be in vitamins. So we need a tiny amount of these, but without them, we have some serious health consequences. So if you imagine a vitamin pill for plants, then what we would also put into our plant vitamin pill is chloride ion or chlorine. They need iron, they need zinc, they need copper, and they need nickel. So some metals, and that, for example, the iron is actually needed as part of the structure of the photosynthetic pigment and other pigment molecules, right? So they just need tiny amounts of these, but they'll have a nutrient deficiency if they don't get them. So one thing I find really interesting about plant evolution and how plants look in different environments, if you're thinking to yourself, that phosphate deficient plant, I have a beautiful house plant that I got, from Home Depot or wherever, that has that neat kind of purple color to it. So we see in plants, especially that come from tropical environments, that may be low light environments. They're good house plants because they can live in somewhat lower light levels. And And they may not be good outdoor plants because they're tropical and they need to have a warmer, moister environment. So if your instructions are to spritz your plant, it might be a tropical plant. There's so many different plant species in these tropical environments. There's so many different insect and herbivore predators on them that some of these plants have, over evolutionary time, adopted a strategy where they're going to pretend to be a sick plant, right? So their normal color is that sort of purpley brown color, and that it's thought to discourage herbivory because the herbivore might think that it's an unhealthy plant, that it's not going to be good to eat. And so we have an evolutionary strategy as well. Pretend you're sick and hope the predators won't eat you. All right, so we're also going to talk about mutualisms, right? Back to our interspecific interactions that we talked about earlier in the semester in ecology. And living in the soil are going to be other organisms that the plant is interacting with. Sorry about this, my screen just went blank. All right, my screen is back again. We'll carry on. And if I remember to slice out this couple of minutes intermission. All right, I hope you grabbed some popcorn and you're ready for more biology. The soil is going to have bacteria in it, and it's also going to have fungi in it. They aren't just sitting there, and they are actively interacting with the plant roots. In fact, we know that plants, bacteria, fungi, when they made the transition to land that plants made in their evolutionary history, that the fungi and the bacteria were already associated with the plants on land when that happened. So plants may not be able to actually live on land without their bacteria and their fungal mutualists, right? So remember, a mutualism is a positive association between two different species that benefits them both, right? We'll see how it benefits the plants, and then I'll ask you to think about what could the plants be doing that would help out the bacteria and the fungi? What do plants have to offer in this deal? Right, so we have all kinds of different bacteria living in the soil. And then the most important thing that I want you to know is that when we look at the atom nitrogen, plants need to get nitrogen, right? They need to have it to build proteins that are going to be used in all parts of their body structures. Plants can't absorb and get into their bodies, nor can animals, nitrogen gas, right? We have abundant nitrogen gas, N2, in the atmosphere, but it has to get mixed down into the soil in order to go through a chemical reaction that is done in the bacteria in order to convert it into what we consider a biologically available. Now, this is very biased, right? Bacteria are living things. So if we said, nitrogen on the whole isn't biologically available, that's just silly, because bacteria can take it and they can turn it into different molecules. However, we usually think about bioavailability from the perspective of animals and plants. The bacteria have to convert it into a molecule that the plants are able to absorb and to use. All right, so there are a couple different processes that are going on. I want you to focus on these three types of bacteria: nitrogen fixing, ammonifying, and nitrifying. So there's two different kinds of nitrogen containing molecules that plants are able to absorb. Ammonium, so when ammonia mixes with water, NH3, it turns into the ammonium ion, which is NH4+. And then we also have nitrate. The nitrate ion is NO3-. And so there are chemical reactions that bacteria can do, that animals and plants can't do, that are going to convert between these different nitrogen molecules. So this process of nitrogen fixing, right? Nitrogen fixing is converting nitrogen gas into ammonia. Plants, when they're doing photosynthesis, are going to fix carbon. So photosynthesis is a carbon fixing process. And so by fixing, we don't mean it's broken. We mean that we're taking it from a gas form into a biologically available molecule, basically. We're converting it from a gas into, in the case of nitrogen fixing bacteria, into ammonia. And then in the case of cellular photosynthesis, we're taking carbon dioxide gas and we're converting it into glucose. Ammonifying bacteria is very simple. It turns non-ammonia compounds into ammonia, and that is based on the biotic action of the bacteria on the organic material, like in your compost pile. The ammonium ion can be absorbed by plant roots. Nitrate can also be absorbed. So we have these nitrifying bacteria as well. And they have a chemical reaction they can do to convert ammonium into nitrate ion. Right, so we have all of these different complicated chemical reactions that bacteria are able to do that are going to produce something that the plants need and can't make for themselves. The soil mutualism with fungi has a lot more to do with the availability of water than it does with nutrients. So plants can't get up and walk to a place where there's more water. Once they start to grow, depending on the environment, there may be a year there isn't as much rainfall. As the plants are growing, they may not be able to grow very fast, close to a good source of water. But they're going to quite readily associate with some kinds of fungi, not every kind of fungi. There are special species of fungi that form these associations. And these fungi are called mycorrhizae. So myco from the word for fungi and the word rhizae for the word for roots. So we have these fungi that become associated with the plant roots. So the hyphae actually penetrate into the tissue of the plants, but they stay in between the cell walls. So they don't actually become fused with the body. They kind of wiggle their way in between the existing structures. The plant is going to benefit because this network of fungal hyphae that extend beyond its root network are going to give it access to locations in the soil where there's additional water and nutrients that it can't reach by itself. Right, so the fungal hyphae are going to be able to pull in additional water and some nutrients. All right, so the soil fungi are going to be able to increase the amount of water that plants can access. What do the bacteria and the fungi get? All right, these are mutualisms. So we know that there must be something that the plant is giving them, right? What does the plant have a lot of? As much as it can make, right? It's making glucose from carbon dioxide, energy from sunlight. So it's going to be able to pass some of the glucose to its fungal symbionts. It's going to be able to pass some of its glucose to those bacterial symbionts. How does it do that? Well, it can pass it directly to the fungal hyphae. It's also going to be secreting or pumping out into the soil around its roots those glucose molecules so that its friendly neighborhood bacteria can have access to them as well. All right, so it's going to share energy. It's going to share food in the form of those glucose molecules. And that's what it brings to the partnership. All right, now we're going to turn our attention to how the plants are able to move water and nutrients around. So here's our cartoon of the basic body of a plant. This is a large tree, but it applies just as well to smaller plants. They need to be able to absorb carbon dioxide into the leaves and release oxygen from the leaves. When we talked about plant anatomy, we looked at those little holes, openings on the surface of the leaves that allow them to do their gas exchange at the leaves. They're going to be obtaining energy from sunlight and producing sugars by the process of photosynthesis. So all the sugars are concentrated in the part of the plant that's doing the photosynthesis. We want to share those sugars with the rest of the plant. So we need a way to move the sugars from the leaves to the roots, right? Roots need sugars too. They need to do cell division. They need to do metabolic processes, all those pumps, those transport proteins that are using ATPs in order to move required substances into or out of the plant. They need energy from sugar. So we've got mitochondria, remember, that are throughout the plant that are going to be making ATPs out of the sugars in order for the plant to use them. All right, so we got to get sugars from the leaves to the roots. We have two kinds of transport structures, two kinds of tissues in the vascular tissues. We have the xylem and we have the phloem. The phloem is the primary way that sugars will be transported from the leaves to the roots. And the way that I remember that is that that F sound of phloem goes with falls. So phloem falls. Right now there is some transport up, right? But the up transport is going to be done in the xylem. Only fluid movement down from the leaves to the roots is going to happen in the phloem. So in order for the leaves to be doing their photosynthesis and also to have enough water in them in general to keep them alive, they need to be getting water. Well, the way the plants get water is from absorbing it from the soil through their roots, right? So we have to have a way for the water to get into the roots from the soil and to move up through the body of the plant all the way to the leaves. Even in the tallest trees, this is accomplished. They do not have any kind of muscle or contractile cells that provide the force. We look at a giraffe, it has a great big heart, and the heart provides enough energy or force pumping on the blood to get it all the way up into the giraffe's head. Plants just use very specialized structures in order to do that without using any kind of muscle action. right? It does not even take them ATPs in order to do that movement, even the tallest trees. Right, now water is going to be getting released from the leaves as well. And so we'll have evaporation of water through the leaves. They need to kind of let some water be evaporated in order to keep pulling more water up from the soil. We talked before about in the water cycle that a lot of water in the environment can be moving through the bodies of plants and released into the lower atmosphere as water vapor. And that process we call evapotranspiration. Right? So like we perspire, we have perspiration. Plants have transpire water. So that's like plant sweat. The reason it comes out of their bodies though is from evaporation. They don't have a mechanism like we do that sort of squirts it out onto the surface. All right, notice what else is coming into the roots from the environment, and that is oxygen. As we talked about before, the roots themselves are not primarily doing photosynthesis. They are principally doing cellular respiration. So they actually need to get oxygen and are releasing carbon dioxide. Leaves are doing cellular respiration as well, but the amount of it is sort of, if you look at the net of how much oxygen they're taking in versus how much they're putting out, there's so much more photosynthesis they're doing that if we're just monitoring the amount of gases coming in and out, it will look like they're just doing photosynthesis. All right, so we're also going to uptake those other nutrients from the soil. All right, so we want to look at how water and materials get from the soil into the roots and up through the body of the tree. And then we're going to look at how it moves down. All right, so when we look at, we zoom in on a plant root, right, R-O-O-T, root plant, we're going to see that each plant cell has a cell wall on the outside. Something you need to know to understand what's happening with how water moves through the plant is that the cell wall isn't a solid structure like a brick wall. It's actually spongy. It has little gaps and spaces in it. And so water can actually seep through. Like when you spill on the counter or the table and you put a napkin down and the water kind of wicks across, sort of goes through the material that makes up the paper. Paper is made from ground up plants. So the same thing happens in the cell walls of living plants, that water can actually ooze through or flow through the cell walls. Inside the cell membrane, there is also this large organelle called the vacuole, the water vacuole. So plants have two compartments that contain watery substances. The water vacuole has a membrane around it, and then it has the cell membrane around the outside of the cell. Because it has these two compartments, it can actually manage the concentration of solutions inside the vacuole versus inside the cytoplasm of the cell. So it can pump ions in between the vacuole and the greater cytoplasm, which is going to influence by diffusion where water is moving, right? So it can force water to move into the vacuole by pumping solutes into it. It can also force water into the cytoplasm by pumping the solutes back out to the cytoplasm, which will cause water to go from the vacuole into the cytoplasm. Right, so the plant is able to manage the concentration of its cytosol and also the contents of its water vacuole. Notice there's an interesting thing here in the way that plant cells are connected to one another, right? So many types of plant cells have actual continuous little tubes of cell membrane called plasmodesma. And those connect the cytoplasm of one cell to the next. We only have a few examples of this in animal biology. So they're actually sort of like megacells that have these connections in between them. So cytoplasm can actually flow in between the cells. So when we look at a little root here extending out into the soil, and we imagine water getting from the soil into the cells and then moving through those cells towards the middle of the plant in order to get moved up into the escalator of the vascular system, it turns out that there is more than one way that a water molecule, if we followed what happened to it, that it could get into the center of the plant. It could just ooze its way through cell walls and never cross a cell membrane and get to the inside of a cell. It could never get into the cytoplasm, right? And so we're gonna call this way, right, a route, like a bus route, not a route like a plant root, a route. We're gonna call this the apoplastic. So that's outside the cytoplasm. It can move continuously through the shared cytoplasm between all of the cells. So that's called the symplastic route. The symplast is this shared cytoplasm that is continuous between all of the different cells. And it can actually go a way where it could pass outside of a cell, into the cell wall, keep oozing through, go back into a cell by crossing a cell membrane. So if it crosses a cell membrane as it's going through, whether it's going into or out of, we're going to say it's gone via the transmembrane route, which is that it has crossed a cell membrane. All right. So the symplast is this shared cytoplasm. The apoplast is the spongy cell wall that's outside of the cell membrane. So if we're the body of a plant and we're taking up water and we're taking up nutrients and we're taking up sometimes other things that might be present in the soil, do we want to have a molecule that has just kind of oozed through the cell wall? Do we want to let that get inside of our bodies? Do we want to let that get into our circulatory system? Probably not, right? We want to be able to screen or control what molecules are going into the rest of the body of the plant. So there's a mechanism that actually forces all the water that's coming towards the middle of the plant root and then getting delivered to the rest of the plant that forces it to cross a cell membrane, right? So when we think about the body, a cell, an organism that is just a cell, or an organism that's multi-celled, somewhere, something has to cross a cell membrane to get into the organism, right? Even when we're thinking about an organism like ourselves, where we're breathing in oxygen, that oxygen has to cross the cell membrane to go into a cell, line the lung before it gets transported into our circulatory system and can get somewhere else. Same thing with our digestive tract. We've got a food molecule inside of our gut. It has to actually pass through a cell membrane, the epithelium of the gut, in order to get into our circulatory system. So we have these screening processes as well. It looks a little different in the plant. So as we're looking at one particular water molecule, it's going from the tip of a root hair and making its way towards the major transport system of the plant, the xylem vessels, these cells, these tubes that are going to take it to the rest of the plant. There is a waxy layer that's embedded in the cell walls in this ring. So if we're looking at the middle of the root, we see a ring of this waxy material. And what it does is it forces any water and solutes that have gotten to that part of the plant, it forces them to cross a cell membrane before it gets into the middle of the main part of the circulatory system. So this is an important quality control step of forcing those molecules to cross a cell membrane. It can be regulated then by the plant. So based on its needs, its homeostasis can be maintained. Maybe it needs some ions, but it doesn't need others. It can stop the crossing of some molecules. It can encourage the crossing of other molecules. It might be a way to protect itself against toxic molecules and potential pathogens as well. This structure, this layer of this waxy material is called the Casparian strip, right? The Casparian strip. And its function is to prevent unwanted materials from entering the xylem vessels in the center of the root. All right, how does the water get from underground up until the top of the tallest tree? We're going to do a little physics and explain how that happens. All right, so when we talk about diffusion earlier in the semester, we know that where we have a higher concentration of solutes, that will have water flowing in that direction. All right, water is going to go down its concentration gradient. So we have this osmosis that's happening. It turns out that there is another force that can affect how water flows. It isn't just the concentration difference. There's also a pressure difference that can be involved. So this series of images walks us through the steps of understanding how osmotic force and how the pressure force can influence what direction water molecules are going to go. And together, this chemical concentration and the pressure differences are called water potential. So water potential has a symbol we're going to use for it, and it's this Greek letter psi. We're sighing because we're exhausted by this lecture already. So we're going to have a value of total water potential, psi sub t, that is shown at the very bottom of each of these panels. And we get it by adding together the water potential that's based on the solutes and the water potential that's based on pressure. So again, just a little bigger, we're going to have a psi sub t, or total water potential. That's going to be the sum of psi based on the solutes and the psi based on pressure. We're going to define pure water at a standard temperature and pressure having a psi of 0. All right, so pure water has a psi of 0, and 1 atmosphere of pressure is going to be defined as 0 pressure as well. So we can have positive and negative values of each of psi of solutes and of pressure. Sorry, we can't have a positive value for solutes. We can only have a negative value because the water is going to follow the solutes, right? So it's going to cause a pull. The higher the concentration of solutes, the more negative the value of the water potential is. Water is going to go down its water potential gradient. Negative numbers are bigger, negative numbers are more negative. Bigger negative numbers are going to pull the water more than smaller negative numbers are. Easy for me to say, right? Okay, so water is going to go down its water potential gradient, whether it's caused by pressure or whether it's caused by solutes or some combination. So we see the water in the first panel flowing to the right-hand side of our little U-shaped tube. We have solutes, we have a certain concentration of solutes, it's going to generate a certain psi sub s. So the total psi is going to be the sum, which is going to be negative, which means the water is going to go that way. So we also have the possibility of putting pressure, right? Pressure, or a push, what we normally think of as pressure, is going to be positive. So we have a push is going to be positive. Push. Right. A pull is going to be negative. We may not think about pressure as being negative, but we can pull, like with a syringe. So where we have, we're sucking fluid into a syringe because we're going to, get our vaccine in there and get our COVID shot. That's a negative pressure, right? So a pull is a negative pressure. We can create a negative pressure in an airspace as well by expanding the volume. So we talk about negative versus positive pressure breathing also when we're talking about respiration in biology. Another word, so we might talk about a push as being pressure. This is a pull being tension. So we're going to define a positive pressure, a push, as a positive value of psi. If we have our unequal concentrations of solutes and our water has moved to one side, we can actually force the water back to the other side if we exert a pressure that exactly equals the isotonic water potential that's generated by the difference in solutes. All right, so if we put a pressure, if we shove on that water, we can actually, if we increase the pressure, we can actually force the water to go back the other direction even. All right. So the third panel shows we have not just compensated for exactly the psi sub s, right, but we have exceeded it. That means we're actually going to force water over into the left-hand side. Right, now the other way we could accomplish the same thing, so on the fourth panel, right, instead of putting a push onto the right-hand side of that U-shaped tube, we're exerting a pull on the left-hand side. My pull and my tension actually apply. I wrote it over the C panel, but it should go with the D panel. I'm trying to, all right. So with these two different forces, it turns out that when we look across the whole body of the plant, from the soil, and the roots, and the stem, and the leaves, and the air spaces inside the leaves, and the outside air, right, all the way over on the left-hand side of this figure, the outside air is going to have the lowest water potential, right? If it's dry air, it's going to have about a minus 100 millipascals measure of water potential. Sorry, megapascals. So when we look across the whole body of the plant, there is a continuous, it has to be continuous, gradient of water potential going from the soil to the outside air. That is going to tend to pull the water up from the roots, and as it evaporates, it's going to be moving from the leaf into the air, and it will evaporate because there is a negative water potential between the tissue of the leaf and of the outside air. The other parts of this system that work to make it happen, all right, not the only, if there was just a water potential gradient and nothing else, then we still wouldn't be able to see how water moves up in the plant. And this is a hypothesized mechanism. The physics and chemistry of exactly how it works is still not entirely understood. So this is our model or our mechanism, the way we think plants do this, more or less, it makes sense. And that is, the plant maintains this, there's a negative water potential gradient from the roots to the leaves. In order for the plant to take advantage of it, right, it has to have, though, a continuous string of water molecules connected all the way from the leaves to the roots. And the reason that can happen is because of the physical and chemical property of water, that it's polar. And water has a little negative end and a positive end, and the positive end of one molecule and the negative end of the other tend to stick together like little magnets. All right, so as water is evaporating from the leaf, it's pulling at the next water molecule behind it, and it tends to pull up from the bottom of the plant. So the continuous chain of water molecules going all the way down. And so that pull is a tension, right? So that combines with the structure of these little tubes, right? So if you've ever tried to use different kinds of syringes, it's much easier to pull a syringe that has a really narrow diameter as the syringe gets bigger and bigger. It takes a lot of force to pull on it. So when we look at the structure of the plants, even in the biggest trees, the tubes, these xylem cells that the water is coming up through, they're tiny in diameter. They're you have to look at them with a microscope, right? If we slice open a body, right, and we see these big blood vessels that are passing through it, they're visible to the naked eye. So there's a much bigger diameter of the blood vessels in an animal than of these vascular tissue cells in a plant. And so the same thing works with the these tiny little tubes. That works when you did a lab in your chemistry or your physics class on capillary action. So we have a little tiny tube, a little tiny glass tube, and it's dry and it's empty, and you tap it onto water, it's going to pull the water up inside of it, right? And so the exact same thing happens in these xylem cells. The reason that happens is because based on the structure of the molecules that are making up that tube, the water can cling to them, right? So it doesn't fall back down again in the plant because it's sticking to the sides of the xylem cells. When one thing is sticking to another thing, a different thing, it's called cohesion. Right? We have a group and we want to have a cohesive or coherent group. That means we're sticking together, right? We're supporting one another. Cohesion are things that are sticking together, they're the same. Adhesion is when we have things that are sticking together that are different, right? If I stick something onto myself, like an adhesive bandage, then that is one thing sticking on something else. All right, so the cohesion, is the water molecules sticking together by hydrogen bonding. There is also adhesion, that is the water molecules are going to stick to the cell wall of the xylem cells, also by hydrogen bonds. And so this allows the water molecules to kind of keep connected to one another, keep kind of crawling up inside of these little narrow tubes by capillary action, as well as by the overall gradient in water potential. Right, so we've got cohesion, adhesion, we have water potential gradient, and we also have a little bit of force that comes from body water in the plant coming down on the phloem side, right? So it gives sort of a little pressure that we'll see in the next slide as well. All right, so we don't have a pump, we do not have ATP, we just have the physical and chemical properties of the cells that are for transport, and we have the properties of the water molecule and of the cells that make up those vascular cells. Pretty amazing. There is a maximum limit on the height of trees that this system works for. All right, it's somewhere around it's over 400 feet, but there aren't 600-foot trees. So we know that there is a limit to the ability of the system to work up to a certain heights of the plant. All right, and so what ends up happening is that water is actually moving up in the plants, right? So we have a flow, and instead of water molecules moving by diffusion, which could happen only over very short distances, we actually have bulk movement of water like a river flowing or the blood flowing around in our bodies. We don't have single cells moving a little ways by diffusion. So when we look down into how the water gets into the roots in the first place, we have sort of this negative potential generated by the rest of the water continuing to move up. The plants are able to also, using their ability to manage solute concentrations in the vacuole compared to the cytoplasm, they can pump a bunch of solutes into the cytoplasm that will increase its solute water potential and pull more water in from the soil into those root cells if it needs to. Right? So the plant can actively manage its water uptake based on its physiological needs as well. Right? They look like they're just sitting there doing nothing. The plants are busily establishing and maintaining homeostasis like we are. All right, one of the ways that they can do this has to do with the stoma on the leaves. Now they want to have the stoma open so that they can get air into and out of their leaves so that they can do photosynthesis. Right. Water, if their bodies are getting dried out and there isn't enough water available for them to keep taking it up, then they can preserve water, not lose water by closing the stoma. So there's a trade-off. They can only do photosynthesis if their stoma are open, but if they're getting too dried out, then they want to close their stoma. The stoma will close automatically if the plant gets too dried out, right? But the plant can also decide to close them, right? How do plants do this? They don't have any muscle cells. How is this regulated, right? They do it by pumping solutes and then the water flows after them. No cell, no living thing can actively pump water. There is not a protein that we have a gene to make that pumps water. What do? We pump solutes. We use ATPs to move them from one side of the cell membrane to the other. And once we've elevated a solute concentration on one side of that cell membrane, the water follows it, okay? So we're gonna move water by pumping solutes and then letting the water follow it, all right? Weird thing about the structure of these stomas. So the stoma is actually the space, right? The space that's in between two cells called the guard cells. The guard cells are kind of sort of U-shaped. When the cells are all full of water, they're rigid or turgid. They have this bent shape. When they become flaccid, that is floppy, they don't have enough water in them, like pumping up a balloon, right? When it's full, it'll be stiff. And when it's not so full, it'll be more floppy. Turgid is a biology word for full of water and therefore stiff. Flaccid is a word for not full of water and therefore floppy. When the guard cells are turgid, the stoma is open. That's just because of their shape. When the guard cells are flaccid or floppy, they flop together and close. So when the plant is water stressed, right, the guard cells will become flaccid and the stoma will close sort of automatically. But if the plant chooses to, based on its physiology, decide to close the stoma, then it can also do that. And the way it does it is by pumping potassium out of the guard cells. So the guard cells have a reserve of potassium. They're going to actively pump the potassium out of the cell. The water will follow. The stoma will get floppy or flaccid and will close. And so that's shown in the lower right hand panel, this one. Right, so we pump, the red dots are representing potassium. The cell has pumped the potassium out, the water's followed, the guard cells have gotten flaccid, the stoma has closed. Right. Reverse is true. It can pump the potassium ions back into the cell. They will become turgid. The stoma will open. Some plants have a repeatable day-night cycle of opening and closing their stoma that is equivalent to sort of wakefulness and sleeping physiologically. All right, so those are structures that are related to how water gets out and into the leaves, or out of the leaves, and how we get water moved up in the xylem. How water moves down, body fluid, plant body fluid, which I should have said before is called sap, right? SAP, sap. So the body fluid that is going down in the down, falling in the phloem, just works by gravity, right? So these little tubes in the foam are a little larger in diameter. That's going to facilitate just the falling based on gravity. Now, what the plant actively is doing is where sugars are being manufactured in the leaves, it's going to actively pump the sucrose or glucose sugar molecules into the phloem. It's going to take the elevator down, and then wherever the sugar needs to go, it will get pumped or actively moved into those cells. Now, what a lot of plants do or have to do is that they're going to have to store up energy reserves for winter. So if we have plants living in a temperate climate, or we don't have really plants in a polar climate, but in a temperate climate where it's going to be cold and snowy and not enough sunlight to do photosynthesis during the winter, they're going to have to produce enough extra sucrose or glucose in order to store it during the winter, so that when they're ready to start growing their new leaves in the spring, they have enough stored energy and enough molecules that they are able to manufacture those themselves. How do we get enough energy to build new leaves when there aren't any leaves on the tree? Well, it has to be stored. So just like a bear adds fat to its body to store during the winter while it's hibernating, plants are going to move all of those sugars and they store them in their roots. They may have storage organs, like we think of a potato or tuber, this large underground plant. But very large trees, they have such a big extensive root system, they don't need to grow an extra special organ. They just have a very high sugar concentration of the sap that's in the roots. In the spring, the sap starts to rise, and so they're moving those sugars up into the tips of their branches in order to grow new leaves. We harvest this in the form of maple trees, this sweet sap, and then it's boiled and concentrated. It isn't as sweet as maple syrup is when it's in the tree, but we can harvest it. We actually harvest it sustainably. We take a little bit, it doesn't hurt the tree, and well, it doesn't kill the tree. And then it's boiled to drive off excess water and make it into an even sweeter, more delicious product. All right, the last section of this lecture has to do with how do plants detect and respond to things in their environment. So one of the things we know is that the plant needs to know which way is down and which way is up. So it wants to grow its roots down and its stem and leaves up. Now, if it can detect sunlight, it will know that that direction is up, but the roots themselves are underground. They may not know where the sunlight is, it's dark. And some roots, not all roots, we don't know exactly how this works in all plants, but some plants actually have a structure. So when we think about the way that an animal can tell if it's upright versus upside down is we saw an example of an organ that works like our inner ear in humans that we have in crustaceans. We have those little statocysts. It's a little spherical shape and it has some little grains of calcium in it. So statoliths are a similar structure that we find in some plant roots. And they are these really dark purplish staining structures. Now they're dense balls of starch molecules. Plants have lots of glucoses. They can make them into dense molecules. So we see that when this plant was first growing, right, and its little root, now the very top panel is how these cells looked when this plant was upright. Right, so the plant is upright. These little statoliths are going to be at the bottom of the cells, right? So the plant knows to grow in that direction, to go down. So we turn this plant root on its side, and after a little time, right, those dense starch grains are going to fall to the side, just like the little lifts do in our crustaceans staticist. And that we'll see now that this plant root, it's going to start to grow in the direction that the statoliths have settled. Know exactly how it knows that, but we can actually see a structure in the body that is causing, we can through experimentation demonstrate that it causes the observed phenomena. Right? It's going to start, cells are going to start dividing more frequently on the side that those statoliths have settled on. All right, so plants can detect the force of gravity, and their response is that the roots are going to grow towards it. Right. So this word gravitropic means that plants have a response. And the roots, they have a positive gravitropic response. That is, they're going to grow towards the thing, right? If we look at the leaves and the stem, right, they have a positive phototropic response, right? That they're going to grow towards light. Now, when we talk about animals, we'll see that we have we don't just grow towards things, we get up and we go, right? And so this tropic response is the plant thing. If we want to talk about animals going in a direction, it's a taxis. Right, so humans, when we're cold, maybe we might have a positive thermotaxis. We might go towards something warm. Roly-polies like to be in moist, dark environments. So they have a positive hydrotaxis, and they have a negative phototaxis, right? They're going away from light, they're going towards water. All right, but our plants have a tropic response. Yet another weird use of this word. All right, plants can do other things. They can respond to mechanical touch. Do we understand exactly how that works? No, we don't. They don't have a nervous system. They have some kind of structure that is not at the tissue level. It is not at the organ level. It is at the cellular level, where they have an ability to cause a change in a cell. It doesn't work just like our neurons do, right? It works, it triggers the pumping of ions, right? Like our nervous system, triggers the pumping of ions, which then rapidly move water, right? So plants work on hydraulics. They can do this thing where they pump water from one place to another, and this structure is a motor organ in the leaves of a particular kind of plant. It's called touch plant or sensitive plant, or mimosa is its genus name. And it's kind of a weedy little plant that occurs in the tropics. And when you touch it, it folds its leaf up. There's other plants in the same genus that not only have this within one leaf, so the leaflets are folding, but they can have it a whole leaf with multiple leaflets on it. So it'll actually drop down. So imagine you're a grasshopper, you land on this plant, you're about to eat its leaf, and boom, you're just dumped off, right? So this is an anti-herbivory adaptation. Same thing with the leaf closing. It sort of flips, it's rapid. It sort of prevents an herbivore from actually sitting on the leaf and eating it. So the other types of movements that plants can make, again, from pumping water onto one side versus the other, is that often we'll see leaves sort of opening up or sort of rising up and then sort of drooping down. Flower petals may close during the night and open during the day. Certain kinds of plants are actually going to follow the direction with their leaves that sun goes. Sunflowers are known for this reaction, that they'll actually orient towards the sun as it happens to move across the sky during the day. They can also, in response to physical touch, they may have particular parts of the body that are going to grow. So we have different plants that are going to grow onto something like a vine. And we have maybe pumpkin plants or other types of plants that are sort of floppy, but they want to grow up onto something. They have these little tendrils that grow out and they'll curl. So when they bump into something, they actually start to grow. They look for a place to grab onto. They sort of wave around over the course of several hours, if you can capture it in slow-mo video. And when they bump into something, then they actually wrap around it. So the plant over a long period of time will grow, it will bend, and it will start to grow its structures towards things that it wants as well. It'll grow towards light typically, and its roots will grow towards water. So these are the kinds of overall responses to things it can detect in the environment. Now, we may see that the plant, it doesn't have a single body organ or structure, a place where the ability to do this reception, like we have eyes, they are responsible for our photoreception. We can't tell light from dark in our fingertips, but it turns out that every leaf of the plant, besides having pigments that do photosynthesis, also may have pigments that detect the light. They aren't taking the energy from it. They're just saying, is it there or is it not there? In certain wavelengths of light, it can tell the difference between them. That's part of the way that plants tell the difference between spring and fall, is the angle of the light coming in from the sun changes. And so the wavelengths of light, the proportion of different wavelengths of light change. So plants are able to detect and respond to a whole variety of stimuli in their environments. And I want that to be one of the take-home messages you have. They're not just sitting there, they're doing all kinds of things, and they're actually actively responding to their environments to protect themselves and to continue doing homeostasis. Plants are under attack from pathogens just like animals are. A lot of them are viral and bacterial. So besides just herbivores trying to munch on their leaves, they actually need to have an immune system which works inside their cells in order to try and detect and manage infections. So they have kind of like animals do, they have sort of a short-term initial version of their immune response and a slightly longer term version of their immune response. Plants do have options that animals don't do in some cases. So we have a plant that's got thousands of leaves on it. It detects a viral attack. There's an infection in that particular leaf. The plant has an option to just drop that leaf off, right? It's going to say, I don't want to get that virus to my other leaves. The best thing for me to do is to sacrifice it. Right? So it's just going to dump that leaf off. So what's going to happen when that pathogen is detected by the leaf? It's going to turn on genes in the cell that is just the local, right? So this is going to be localized to the place where the virus is actually attacked. And it's going to secrete antimicrobial proteins, and it's going to initiate cell death in a regulated way in that leaf, right? So it's going to kill the leaf for the good of the plant. Now it's also going to release kinds of molecules that act like hormones that are going to go out into the rest of the plant and are going to let the rest of the plant know that there is this dangerous virus nearby. That's going to turn on genes, right? So signal transduction pathway means that the information is going to be turned into a response, which is that genes are going to be turned on and that the production of those antibiotic molecules is going to get started and ramped up in the whole rest of the plant, right? Every living thing has body processes that it can do in response to a threat or a danger. It doesn't want to do those all the time because it takes energy to do that, right? It takes energy to generate those antibiotic molecules, so it only wants to turn them on when it needs them, right? So it's going to turn those genes on when it gets a signal that it is being attacked by a pathogen. And then that should help protect the whole rest of the plant if that pathogen is nearby. The other thing that can happen is that if it has those molecules on all the time, then it will be easier for that pathogen to evolve to be resistant to them, right? So we have disease resistance, we have protection from pathogens, just like predator-prey evolutionary arms race. We have that going on across all species, across all kinds of organisms since the beginning of time into the future, right? So we have this coevolution between hosts and pathogens as well. All right, our last set of slides has to do with plant hormones. So just like animals, plants have a variety of different molecules that they use as hormones, and they have those, they can secrete them in different cells. So we don't have the equivalent of endocrine glands, but they are able to secrete these hormones in certain cells, and then they will get transported across the rest of the body. The presence or absence of these hormones and also the relative proportions of different hormones are used as messages that different parts of the body know what they're supposed to be doing. These are used to communicate things about how to respond to the environment, for example, growing towards or away from a resource. Also to determine what part of the plant the cell is in so that the cells differentiate into the right kind. So hormones are involved in the plant knowing which end is its stem and which end is its root. And also they're involved in processes that regulate development, just like in animals. Really neat experiment that was done pretty early in biology history to demonstrate that hormones exist in plants. And so it's a nice example of a well-designed experiment. And so we have these different treatments that were tried in order to see, suppose there is a hormone molecule that is in the tip of a plant that exerts a function on how the plant grows, right? How could we tell that, right? Well, we'd have to set up an experiment where we could get that material, whatever that is, suppose there's a hormone. How do we get the hormone out of the tip, right, to do a really controlled experiment? Well, let's cut the tip off and let's put it on top of a little square of agar, right? That's that watery matrix, sort of a gelatinous substance we used in our agar settling plates earlier in the semester. So if there's a molecule that's water soluble, then it'll diffuse into this agar cube. So it's shown in pink, these hormones. Suppose they exist, right? Suppose they're water soluble, we transfer them into the agar cube. We have a bunch of these agar cubes. We have a whole bunch of replicates. We have plants that we've put into these different conditions. We've assigned them randomly to our treatments and controls. And we want to see, A, if there's an effect of cutting the tip off the plant in the first place. Do they just all die, right? We want to see if the agar itself could cause anything. We want to see if the material that's transferred into the agar cube causes growth in general. And also if it's specific to certain cells. So if we put it on some of the cells on the tip of the plant, does the whole tip grow, or does the only some of the cells grow? Right. So there are these 5 treatments, which include 2 different controls. And so these little agar cubes, they were set on the tips of these plants. And what happened was the one with just the tip cut off didn't grow. The one with just the agar didn't grow. The one with the tip, the square put right on the top, across the whole top, it grew straight. And then when we put it just on one side of the tip, I don't know how they did this, but only the side that has the agar cube grows, right? So we can see that if a hormone is only released sort of onto half of the tip, then that is a mechanism that the plant could use to grow towards one side, right? So this tipping, this growing towards, we find a mechanism for how. So one, we conclude, yes, there are hormones in plants, and that they regulate growth, and that they can also cause this phototropism that we see. So one side of the plant is in the sunlight, the growth hormone is only released on the opposite side. It causes it to grow towards the light. All right, so it turns out that the name of that hormone, the one that stimulates growth in the shoot tip, is called auxin. And it isn't just one auxin molecule. There's a whole family of auxin molecules that are slightly different from one another. They have slightly different functions, but generally we can put them into this category. Like we have steroid hormones, there's a bunch of different ones, right? So saying auxins is a way of summing up a group of molecules and what they can do in the plant. So this auxin molecules are secreted by the shoot tip. And if we, and they trigger elongation in the shoot and the roots, right? So they tend to stimulate growth. And another thing that the auxins are responsible for is whether the plant's going to keep growing just at the tip or whether it's also going to start growing little branches off to the side. In many plants, if we pinch off the main tip, it will start to grow out to the sides instead. So it's basically like, whoa, I didn't want to grow that direction. I'm going to grow somewhere else. Some plants can't grow to the sides. If we pinch off their tip, they die. And some plants will grow a lot to the sides no matter what we do, right? But this main shoot tip tends to be across all plants. It tends to be the dominant direction that plants are going to grow. And so we refer to this phenomenon as apical dominance, right? Now, if you want your plant to be shorter and fluffier and bushier, pinching off the tips is going to be a way to do that. So basil, for example, you might want to make it grow more to the sides rather than growing taller. You pinch off some of the tips and you get more leaves from your basil, for example. All right, so it turns out that auxin is also involved in the process plants use for telling what end of the plant is the stem end and what end of the plant is the root end. All right, so auxins are released at the apical stem end, the apical meristem. So if you're a cell that has a high concentration of auxins around it, you know that you are a stem cell. And there's another class of molecules called cytokinins. Cytokinins are released from the roots. And what they do is they trigger cell division, right? So the roots, which know what resources are available, are going to release the cytokinins when it is a good time for the plant to be growing, for the cells to be dividing, right? So cytokinins from the roots trigger cell division, auxins from the shoot trigger cell elongation. And the ratio between them lets the cells in the plant know whether they're closer to the shoot tip and to become a shoot tip cell, or whether they're in the root, right? High concentration of cytokinins relative to auxin, you know you're a root. If they're about half and half, you know you're a stem. You're somewhere in the middle. All right, so interesting stuff. Let me write down that word so that you can see it because it's not on a slide. Cyto. All right. Our next group of hormones are called gibberellins. Gibberellins is shown on this slide in the top left-hand side, that word. And gibberellins are involved in seed germination. So when the seed detects that circumstances are appropriate for its growth, releases gibberellin. The gibberellin triggers release of other hormones and enzymes that start the processes that make plants complete the process of germination, right? So it's a trigger, it sets off other processes. Gibberellins are also promote fruit growth. And so when we see not just inside the seed, but also in the ovary of the flower, that they're going to help be responsible for growing the fruit. Ethylene, yes. C2H6 is another plant hormone. And this one is released when the plant is under stress, right? We have stress hormones and they trigger a whole bunch of different responses. And plants also have environments that are stressful and they have a stress response, right? Look at the plant on the far left that has 0 exposure to ethylene, and the plant on the far right that has the highest exposure to ethylene. And you can tell the difference between the plant body that's grown up in this stress response environment compared to a normal plant, right? So there's 3 different things that we see that are part of the plant stress response. In fact, it's called a triple stress response. One of the things you should be able to notice is that that growth slows. The shoot doesn't get as tall, right? We're in a bad environment, maybe growing up isn't the smartest thing to do. Some of those resources are redirected, right? Notice also something about the stem is that it doesn't grow as tall, but it grows thicker. So it thickens and reinforces the stem. This happens to plants that are trying to grow where there's a high wind a lot of the time, right? You see that they aren't as tall and that they're thicker. They have physically changed their growth form to be able to survive in that environment. Notice the other thing that happens that's really funny, right? The plant didn't grow straight up, it grew to the side, right? So I noticed this in my yard when I go outside in the spring and the weeds are starting to come up. They're starting to come up in my yard now. I've got some rocks laying around. You pick up a rock and there's a seed somehow that got underneath it and it germinated. And what's happening to those plants? They're still growing. They tried to grow up, they hit the rock, they took a 90-degree turn and they're growing out to the side. So they're trying to grow out from under that, that stressor that they're encountering, all right? And some of them make it, right? So they're going to turn to the side and they're going to try and avoid this stress, right? Horizontal stem growth. Another really crazy thing that ethylene is responsible for. Remember we said gibberellin is responsible for fruit growth. Ethylene is responsible for fruit ripening. So we as humans have taken advantage of this. Bananas, when they're ripe, don't last very long and they're soft. So we wouldn't want to transport bananas when they were ripe because they would not get there in time before they got mushed. And we can't pile a whole bunch on one another when they're ripe because they would smush and we'd get bruised, icky bananas no one would buy. Turns out that when scientists discovered that ethylene promotes fruit ripening, you can actually take a whole train car of bananas, ship it in a container that does not have any ethylene in it. The plants have been suppressed from secreting ethylene. They're harvested too early so that they haven't started secreting it themselves. They're green, they'll stay green, firm. You can stack them all on top of one another. You get them to, outside the doorstep of Costco, you pump ethylene gas into that container and they start ripening, right? And so they will continue to ripen. Not all fruits will do this. Some fruits don't, but bananas definitely do. They start producing ethylene on their own once they start to ripen. And then you can actually take a banana and stick it next to that pear that isn't very ripe or that avocado, and it'll actually help it get ripe. All right, our last slide is that we want to talk about plant pheromones. Plants are able to communicate not only with each other as individuals, but with other organisms. There's a class of chemicals called volatile isoprenoids, or VIPs, very important proteins. They're not proteins, they're isoprenoids. And plants will secrete them when they are under stress, if it's too hot, if it's too dry, and they act as antioxidant molecules. So they'll secrete these molecules and that'll help protect the cells from that stress. They also secrete these molecules when they're under attack by herbivores, and predators of those herbivores will be able to smell these pheromones and will be attracted to the plant that is secreting them. So basically, there's coevolution between the plant and the insect predator. Mostly they're insects. So for example, a ladybug can smell that there's a tree in distress, that it has a food source for the ladybug, and so it will be attracted to the plant. There's all kinds of complicated relationships that the release of these chemicals can have as well. They can let a neighboring tree know that there's a pest in the area. That tree can start secreting anti-herbivore molecules that are similar to this immune response they have within their own tissues, right? So there's all kinds of signaling and communication in between plants and the predators of their pests. Plants also talk to one another. They transmit information. They can actually transfer materials to one another, even across materials, across species, underground in their roots. So there's this incredibly active area of plant research that's involved in inter-plant communication, whether it's of the same species or of different species. And so I hope you are a lot more interested in plants than you were when we started this lecture. I think their physiology is fascinating, and there's still a lot that we don't know about how it works.